What is enough? 1 day is enough.
Okay.
I woke up and the world's already worse.
So let's start class. I'm not a happy camper today.
Are we all?
Anyway, that's all I have to say about it. I said in my statistics class, I really hope I can stop talking about this jerk, and because of statistics and sampling and stuff, and evidently that's not the case. In case you're wondering, I'm not happy. I didn't vote for him. I'm surprised. I literally didn't watch any news yesterday. I found out this morning. My wife told me. I was like, so that's the only good thing is that I found out bad news from her because I don't want to hear anything. But let's talk about something more important, more relevant to us, and that's this class. We're done with Unit 3, and let's talk about what where we're headed, and then we'll talk about sort of what we experienced with the Exam 3. So first of all, today's Wednesday. We have normal lab this week, and we're talking about the nervous system and spinal cord. Monday, we do not have school. That's November 11th, so that's Veterans Day. So you have a 3-day weekend. So you have 4 days this week and 4 days next week. On the following Tuesday, so not next Tuesday, the following Tuesday, we have a quiz, and then the following Monday, we have the 4th exam. So that is right before Thanksgiving, and then, so that brings us right to there. And then when we get back from Thanksgiving, we have a quiz and an exam. So we get back from Thanksgiving, a couple of days talking about the brain, we'll have a quiz. The following week, we have normal stuff here, so we're going to go through 2 more chapters there in the following week. And then Monday, December 16th, we have the last exam. If you just look at this here, there's that week, we won't have normal laboratory, but I will be in lab and I'll make it available to you because there's a final laboratory practical. That final laboratory practical, which is part of the laboratory, not the lecture, that final laboratory practical is 30% of your grade in laboratory. So that's a huge chunk. So that means if you're doing poorly in lab, you can really raise your grade. If you're doing well in lab, you can really lower your grade if you really want to. So that's a huge chunk. And that's a cumulative laboratory practical, starting with the first week of class, all the way to that laboratory right there. So everything, okay? So there's still a lot of work to do. And are there any questions about that?
Wait, everything, the lab is everything?
This final laboratory practical is everything. It's everything.
Oh. How many questions are you gonna ask?
So there's 60 questions, 20 on bones, and 20 on muscles, and 20 on
Holy moly.
the nervous system. So there's, and it's in bright space, and it's,
Okay.
so.
Oh, it's on thing?
Yes.
Oh, okay. I thought it was, yeah. I thought you were gonna ask.
No, that would take me forever in a day, and it was no, I can't do that.
Oh, you're talking about like the...
So that's a cumulative final exam. And let's see. So let's talk about exam 3. Remember too that exam 3, the end of exam 3 is this Friday for any new content for the LT assignments. So that's going to close then. So you look at me like, so the due date should have been before the exam, but I delayed it till Friday this week. So that's the summary. I don't remember the other 3. Just check LT for those. But in terms of, I looked at the grades and I compared them to exam 2, which were pretty good. Out of you guys, so there's 5 of you that did about 5 points or so better. 3, 4, or 5 points or better. 3 of you are about the same, a couple points above, a couple points below, between the second exam and the third exam. 19 of you declined. So look around here. 19 of you declined either a little bit or some pretty significant declines. Some of you started off well, really well, and so that decline isn't a huge thing. Others of you didn't really dramatically dropped off. So this is the third exam. There's 2 more, and they're all equally weighted. So if this is the last, if this is a poor performance on your behalf, on your part, then use it as basically motivation. Essentially what we're talking about here is 1 more month, because it's early November. Right? It's November 6th, so we're talking about December 16th with a couple of breaks in between. So 1 more month of stuff, and then you're on your way. Right? You're free from all of this stuff, at least this semester. Okay? So that's what I always try to tell my kids, my biological children, that classes start fast and then
They end faster.
they end fast, right? So let me see here. So just like I've done in the past, we're gonna do a summary of anticipate what's to come. And so I'm gonna switch devices here. So while I'm switching devices, are there any questions for me about what is due, when it's due, or anything like that?
They're still counting. Dr. Clancy,
Yeah.
is this unit harder than the unit before this one?
It always gets harder, Douglas.
I have a feeling that this unit's easy.
Don't say that now. Don't say that now. You're gonna jinx us. You're gonna jinx us.
Doctor? There's a...
So John, how much money you got in your pocket? Let's see here. You got any credit cards here? Hey, can I look at your old picture? So the security guard.
Fresh out.
So the question was, what is this next exam harder than the first 3 exams? So the way that if we were down the street, if we were on the main campus, it would be, because what it would be all of the nervous system stuff, all in one exam right before Christmas. That's 5 chapters or 6 chapters, depending on how you break it out, and it's all would be one exam. What I do is I break it up into 2 exams with 2 quizzes, and so it's smaller chunks. The one thing about it is that will become apparent is that the material that is in the end in exam 4 is definitely overlapping with the material in exam 5. So that's one thing that you have to recognize. So in terms of the material itself, the material itself is definitely more dense. So we'll talk about, after I do this drawing and take these notes, we'll definitely talk about what the vocabulary looks like. So in terms of the quiz for this next quiz, which is quiz 7, we're just talking about nervous tissue. So let's talk about the nervous system, sort of classifying the entire nervous system here. Let me see here. So in terms of the nervous system, you know the basics of it, right? You know that there's the CNS and the PNS. The CNS includes the brain and the spinal cord. the peripheral nervous system has three things. There's this called the somatic nervous system, or what we're going to do is sensory nerves, motor nerves, and I'm distinguishing the term nerve from neuron. They're not the same thing. So there's sensory, there's motor, and then there's also something called the autonomic nervous system. And this is part of the peripheral nervous system. We're going to be talking about the spinal cord in this unit, and then we're talking about spinal tissue or nervous tissue. In terms of nervous tissue itself, what are we talking about here? We're talking about two types of cells. There's neurons and neuroglia. And first thing I just mentioned this a second ago. Neurons and nerves are not the same thing. They're very, very different, and you need to recognize that. Neuroglia, we're only going to be talking about a couple of different types of neurons, but they have essentially the same type, the same thing. So we're going to just say there's only one type of neuron, but there's 6 types of neuroglia. In terms of, so when you have, there's nerves that are part of the sensory nervous system, there's nerves that are part of the motor nervous system, the peripheral nervous system, and then there's autonomic nerves. Nerves that come off of the brain are called cranial nerves, and nerves that come off the spinal cord are called spinal nerves. Regardless of whether the nerves are off of the brain or the spinal cord, they're part of the peripheral nervous system. And anything that is found within the central nervous system is a different type of classified system. When we talk about information here, when we're talking about information or signals, so there's electrical impulses. If you think about all of your memories and all of your brain, everything about your brain, every memory that you have is simply a group of electrical impulses. And it really, when I start to think about it, is really weird that we can remember things simply by, where the hell do we keep our memories? It doesn't make sense, right? But at the end of the day, it's just electrical impulses. So now, if we talk about information, the vocabulary now is going to start to get a little bit more significant here. When we talk about information flow, it either goes to the CNS or it comes from the CNS. If it's not in the CNS, it's in the peripheral nervous system. A signal that is going from the CNS to the PNS is called an afferent signal. And a signal going from the PNS to the CNS is called an afferent signal. The sensory tissue, just in general here, that is sending signals from the peripheral nervous system to the CNS via the afferent pathway. These are called sensory neurons. Or sensory nerves. And ones that are, here, let's just, wait a minute, let me change that. I want to be more specific here. So there's sensory nerves, and information going from the CNS to the PNS are motor nerves. So now the terminology that I'm using now is, so the motor nerves run in an efferent direction, sensory nerves run in an afferent direction. Now, in terms of the neurons themselves, there's different functional neurons. So now this is, I was just talking in blue about the differences in nerves. Now talk about the neurons themselves. So there are sensory neurons. There's integrating neurons. And then there's motor neurons. Once again, the distinction between nerves and neurons are different. There's definitely that, but the information always goes from left to right in this diagram. So the sensory neurons will send signals to the integrating neurons. The integrating neurons will send signals to the motor neurons, and oftentimes the signals that are sent to the motor neurons then come back. Those motor neurons then send signals to the sensory neurons, who get information, and then they either stop something or they start it. So this is talk about information flow, and at the end of the day, when we're talking about different types of neurons here, the shape of neurons really matters. The structure of neurons really matters. And so what I want to do now is draw parts of 3 neurons, just like we drew parts of 3 sarcomeres. So we're going to draw parts of 3 neurons, and we're going to be talking about afferent neurons, efferent neurons, and integrating neurons. So are there any questions about this? So this basic stuff, okay. So, let's see here. If you remember what I drew a cell to look like, it was a blob with phospholipid bilayers, and those phospholipid bilayers had that structure here. We're still talking about that same phospholipid bilayer, but that blob, that circle that I drew as a cell here, is now much more complicated. There's what are called cellular extensions, and if there's numerous cellular extensions, there's one group of one term for it, and if there's singular cellular extensions, very large cellular extensions, that's another group. So the idea here is, let me see. So the cellular extensions that are going to the right is a singular, very large cellular extension, and the central part of the cell itself is this big blob that I'm drawing right now. And these small, numerous cellular extensions, which I'll just do 3 of them, are receiving areas. So these parts right here are receiving, these are called dendrites. Now, in the in the muscular system, we talked about the neuromuscular junction, and I talked about terminal axons right here. So this is called an axon right there, and then there's that's the central part, and then there's telodendria and terminal axons. on the right. So the axon is the central thing, the central process right here. The telodendria is a branch off of that, and then the terminal axon is a branch off of that. I didn't draw on the right-hand side more branches. We have a good cell right here. So there's a very, very substantial nucleus. We have ERs, or endoplasmic reticulum, lots and lots of mitochondria. And you know what those mitochondria are doing, because those mitochondria need oxygen. All they're doing is making ATP. There are inclusions here, and those inclusions get sent down the cell down the axon, along with the mitochondria, and that process is called axonal transport. The narrowing of the main part of the cell is called the axon hillock, and that major part of the cell is called the soma, or cell body. There's nucleus and cell inclusions. That's what I have in this little black thing right here. And then this, let me see here. This specialized endoplasmic reticulum is called a Nissl body. And we're going to look at pictures or stained slides of neurons. And this stained slides of neurons are going to have lots and lots of Nissl bodies. And what Nissl bodies do with those endoplasmic reticulum, they do a lot of cell metabolism. They're building things. They're putting it in these cell inclusions right here. And they're also helping produce other things that help maintain the structure of the neuron. So this is a basic neuron right here. I'm going to add, so this is a single cell. Now I'm going to add to this part right here an additional cell. Right here, there's also small, narrow things right there that are integrated into the neuron itself. These little narrow structures, and those narrow structures they're called neurofibrils, and that's basically helping the neuron maintain the integrity of itself. And I'll label that in 1 second, right here. But this blue thing that oops. And I think if we look back at the picture of the muscle and the neuromuscular junction, that I drew this blue stuff surrounding the axon, and I called it neuroglia. These are indeed neuroglia. So now, so the neuroglia are a different type of cell, and depending on where this neuron is, if it's in the central nervous system, it's one type of neuroglia. If it's in the peripheral nervous system, it's a different type. So I have the general term for this, that whole structure here, this whole projection to the right is called the axon. But the cell membrane of the axon itself is called the axolemma. And then there are these nodes that are found within the area here. There's spaces between the blue cells. So those spaces are called nodes. And nodes of Ranvier. In terms of the signal itself, in terms of what the neuron does, the only thing that the neuron does is it detects something on the left and it sends it to the right. So it sends the signals that electrical signal is going from the left to the right. The electrical signal starts at the axon hillock and it ends at the terminal axon. It ends right here, right there. And that is called an AP, or, whoopsie. So an action potential is sent by definition from the axon hillock to the terminal axon, right here, and then stuff happens to the right. This action potential has, in order for it to get the signal get sent, it has to be ready to send it. If it's not ready, then it's not going to send it. That term for the readiness of the neuron to be prepared to send that signal is called an RMP, or resting membrane potential, and that resting membrane potential is defined as being negative 70 millivolts. So that resting membrane potential is the definition of when a neuron is ready to send an electrical signal. The analogy that I like to say here is that when the neuron is ready to send a signal is like when I'm in my basement setting mouse traps. So every winter, it's getting better, but every winter we have mice in our basement. I don't know how the hell they get in the house, but don't tell my daughter because she doesn't want to hear about this. And so I go and I set mouse traps. Put a little bit of peanut butter on the mouse trap. I set the mouse trap, and then I put it in a couple of different areas, right? And then I go back and bam, it's ready to send that signal, and bam, it kills the mouse. What? How often? Probably like 4 or 5 a year. So I just found 2 this weekend. They were a little crusty. They were pretty old. But with this, every time it's cold, the mice start looking for warm places to be. And once again, I still can't figure out how the hell they get in the house. So that, with all of that work that preparation that I did for setting the mousetrap, I bought the mousetrap, I bought the peanut butter, I put a little bit of peanut butter on the end of the mousetrap, then I pulled the mousetrap back and I sent the trap itself, and then I put it down gently. That is all the resting membrane potential. If I don't do that resting membrane potential, if I don't do that necessary work, right, then the mousetrap can't snap. If this resting membrane potential isn't maintained, then a neuron can't send its signal. That resting membrane potential is maintained by, have you ever heard of the sodium potassium pump? Yeah. So we started talking about the first week of class, and that is determined by, the sodium potassium pump. So there's a lot of information here. In terms of the me see here. I think what I want to do is to draw a bigger teledendria or terminal axon, and I want to show what's going on there. Yes, sir.
What's it called? The fibrils? You forgot.
So, those are called neurofibrils. I'm running out of room here. Let me see. That's a Neuro, neurofibril, and a neurofibril is a cytoskeletal element built by the missile bodies and then sent down the axon through axonal transport so that it maintains the integrity of the axon itself. Is there a question? So there's that, okay? So now what I want to do is to draw a couple more things. So first of all, is this basic cell okay? All right. So just give me a second to erase some things. Just want to take some of these red lines out. If I was better at this, I would move the cells closer together, but we're going to leave it like this because I don't want to spend hours trying to get this perfect. So when you have the meeting of the axon of one neuron and the dendrite of another, that's called a synapse. An example of a synapse, of a specialized synapse, is called a neuromuscular junction. Now, in this case here, this would be an afferent neuron, this is an integrating neuron, and this is an efferent neuron. So this would be a motor neuron. Over here to the right would be a neuromuscular junction in this drawing if this was a motor neuron. This is a sensory neuron. It might be in your skin, it could be in your anywhere in your body, and it's sending signals in an afferent direction. This neuron might send signals to the spinal cord and then to the brain. This neuron might be in the brain somewhere. It might be in the spinal cord, depending on where we're talking about. And then from there, from the brain or the spinal cord, it's going from there to the motor neuron. So there's afferent, integrating, and motor neurons. We have this synapse right here, and that synapse, I'm going to make this bigger. I'm going to make this blob bigger right here. So these sort of clubs right here have inclusions, and they have mitochondria. And now these are the terminal axons. This is a synapse. There's the synaptic cleft. The synapse and the synaptic cleft is the space itself. So synapse is a general term. It just says the space itself is the area between the two cells. In this case now, there's an electrical signal coming down the axon. That AP is coming down the axon, or that action potential. It's coming from left to right. It's hitting the terminal axon, and then as a result of that hitting of the terminal axon, there are these things in here. And we talked about one example of them in there. That's called the material that's found in these cell inclusions that are built in the soma, that are built right in here, and are sent down here through axonal transport, and then reside here, and that's over here. So this is a cell inclusion that is one example of which we talked about already was called ACH, or acetylcholine, and that's a neurotransmitter. When we get to the brain, there's going to be a whole bunch more neurotransmitters that are involved with this, that do specific things, and some of them can be excitatory and others can be inhibitory. When you're talking about the term synapse itself, there's going to be the PSP, postsynaptic potential, or presynaptic potential, presynaptic axon, or presynaptic terminal, postsynaptic dendrite, or postsynaptic neuron. All that means is if we're to the left of this drawing right here, if we're over on the left, it's presynaptic. If we're over here on the right, we're on postsynaptic. So now there's a couple of different terms for that. Postsynaptic. It could be postsynaptic neuron. It could be postsynaptic dendrite. Okay. The dendrites are always on the postsynaptic part. The axon or axon terminals are always on the presynaptic part. So there's presynaptic and postsynaptic terminology that you'll see and hear me use. When the action potential goes from left to right, that signal then changes the resting membrane potential, so that resting membrane potential then causes the ACH, or just in general here, the neurotransmitter through exocytosis to release from the terminal axon. That terminal axon releases all of those, that ACH across the synaptic cleft. It releases it from the presynaptic axon into the synaptic cleft, and that neurotransmitter then goes to the postsynaptic dendrite. That postsynaptic dendrite then has receptors. Those postsynaptic dendrite, those specialized receptors will then respond or not respond. It can either respond in a very specific way, or it will just, and in one example there, if there's enough as a signal, so that if there's a mouse in my basement and it goes up to one of the mouse traps and it just sniffs it and just walks away, nothing happens. So there's an example where there's not enough signal, right? So now another one comes up and it's hungry and it starts chomping on the peanut butter, and then bam, right? That signal, when it was chomping on the peanut butter, that signal was enough to cause the neuron to send a signal. So if the signal is great enough that is arriving to this dendrite right here, or to these dendrites, then the middle axon, this axon, or this middle neuron then starts sending a signal. Okay, and that, again, that action potential is on the left-hand side. There's an action potential or resting membrane potential. And there's a resting membrane potential here. So whenever there's a neuron around, as long as it's a normal, healthy neuron, it's got resting membrane potentials, and that resting membrane potential is equivalent to snap a set mousetrap. Once again, that negative 70 millivolts. Okay. When you're talking about leaving the material, the neurotransmitters leaving from the terminal axon, you're talking about exocytosis. When it's arriving at the postsynaptic dendrite, here, then it's endocytosis. So there's presynaptic. So each cell will have a presynaptic area and a postsynaptic area. So its postsynaptic area will be anywhere from the soma to the dendrites, and the presynaptic area will be the axon, the terminal axon, the telodendrite. And this will continue on as long as there are neurons to receive the information. So integrating neurons come in a number of different varieties. Peripheral neurons, so integrating neurons are part of the central nervous system. Peripheral neurons come in one example. So peripheral neurons are multipolar. Integrating neurons can be multipolar. So I'm just going to write multi. They can be multipolar. They can be anaxonic. And then other shapes too, but those are two examples of shapes that are really, really unique. So a multipolar neuron is defined as is most abundant type of neuron in the nervous system. The multipolar neuron is defined as one with many, many receiving areas, dendrites, and a single axon. So that's a multipolar. Anaxonic neurons are neurons when they're investigated, when they're looked at, they look like stars, and all of the different projections, cellular projections, are equal length. So you can't look at when you're looking at an anaxonic neuron, you can't say, oh, that's an axon and that's a dendrite, because they all look the same. So integrating neurons tend to be very short. They tend to have a lot of, well, there's definitely neuroglia around, but they tend to be very short, and they don't send signals very long distances. So these things generally here are short, and they send signals short. Wait a minute. They're also known as gray, or they appear to be gray. When a neuron has a neuroglia that is wrapping the axon, then it appears one color. When the neurons have neuroglia that, or do not have neuroglia wrapping the axon, then they're gray. The neurons that are short, that are gray, are uniquely found or only found in the integrating system or the central nervous system. So there are gray neurons found in the brain. There are no gray neurons found in the periphery. There are white neurons found in the periphery, and also uniquely only in the periphery, and they're also found in the central nervous system. So the shape of the neurons then is really instructive as to where they are located. Peripheral neurons are always white. And those neurons, because they're white, they have neuroglia that are wrapping the axons, and those neuroglia help send signals very, very rapidly, and it might be hundreds of miles an hour equivalent. And it sends it very, very rapidly, and it also helps maintain the integrity of the neuron. So now I want to talk a little bit about the distinction between neurons and neuroglia here, and then we're going to talk about the action potential, what graphs look like for action potentials. So the neuroglia, there's 6 types. I said this already. Even a different shape of a neuron, at the end of the day, it does the same thing. It sends the signals from the dendrite to the cell body to the axon. And when I say cell division, I mean mitosis and cytokinesis. So when I was talking about the differences between neurons, their nervous tissue, when I was talking about nervous tissue back in chapter 4 in the first unit, I said that nervous tissue does not divide or do not go through mitosis and cytokinesis. And I was talking about neurons. I wasn't talking about neuroglia. Neuroglia are found 4 types in the not in the PNS. 4 types are in the CNS, 2 types are in the PNS. And you need to know all the different jobs of all of these different neuroglia. And those neuroglia then can come in a variety of shapes. And they have very, very specific jobs. So let me just try to save this. So I will post all of this later today. So the resting membrane potential of an axon is negative 70 millivolts. And the way that it looks is that we have an x-axis, or a y-axis rather, and an x-axis. The x-axis is in milliseconds, And the y-axis is in millivolts. There's a characteristic sort of process that, so this, when the neuron is set at about -70 millivolts, it is set to be having a resting memory potential, a certain resting memory potential. And there's a series of areas, and this is what the graph looks like. Now, this is a span of a couple of tenths of a millisecond, so this is not very fast at all. I mean, not very slow at all. So this is the graph itself. So there's characteristic spots here. First of all, let me see here. That green point right here is the trigger. In the first stimulus, so here's two mice that come to my mouse trap. The first one sniffs the peanut butter and walks away. The second one sniffs the peanut butter, takes a big old bite, and it exceeds the threshold potential. That's the green. So so this area right here is called the threshold. When it exceeds that threshold, then the trigger is set here. The trigger is released, and that trigger then this series of events that happen. This right here is called so there's once the threshold is exceeded, whatever that threshold is for that particular type of neuron, the axon of that neuron undergoes depolarization, and then things switch. It undergoes repolarization. Most of the time, it goes too far, and it undergoes hyperpolarization, and then the cell returns itself to the resting memory potential of about -70 millivolts. Once again, this is millivolts here. When we're talking about this, this whole process right here, the only way that this resting membrane potential here is achieved is that you have the sodium-potassium pump. Does anyone remember which ion gets sent out of the cell and which ion gets pumped into the cell?
Potassium goes into the cell, right?
Potassium goes into the cell, sodium goes out of the cell. So why is sodium so bad for you in your diet? Sodium will give you high blood pressure, right? One of the reasons why sodium is so bad for your diet and for you, if you have too much of it in your diet, and give you high blood pressure, is because your entire body, every one of your cells, if it's involved in setting up resting membrane potentials, is sending sodium out of the cell too. Where does it go? It goes into the plasma. So then if you're eating sodium as well and you're adding to your diet, sodium to your diet, then that just elevates it so much higher. And so that's one reason why that's bad. So then what happens here is sodium is in the ECF. sodium is in the ECF or the plasma. What is ECF? Extracellular fluid. And where is potassium? Now, we're talking about at the axon level. So potassium is in the axoplasm. What is plasm? It's the cytosol or the cytoplasm of the axon. So once the stimulus is exceeded, or the threshold is exceeded rather, depolarization happens. And so depolarization happens here. Sodium is imported into the cell until it goes up to about positive 30 millivolts, and then the opposite happens. Potassium is ejected from the cell. And then hyperpolarization is when too much potassium is sent out of the cell, and so then it's returned to resting membrane potential, or RMP, right here, when the level of potassium is appropriate. Right? So it's returned to the cell. Now, this is a single action potential. And this one action potential happens at one spot in a neuron. Happens one spot in an axon, and then if the action potential then propagates down the axon here. So this is one action potential at one spot in an axon, and then right next to it, another action potential happens, and another action potential happens, and another action potential happens. So that there's neurons that are there's white, I talked about white neurons and gray neurons. So there's gray neurons, gray axons, and white axons. And when I said white axons, they have neuroglia that are wrapping the axons, and the gray axons do not have neuroglia wrapping the axons. So that in terms of the, when you're talking about gray axons, these are in the C and S right here, and these are very short. And these have continuous action potentials. A continuous action potential is very slow, and it sends signals short distances. White axons can happen in the central nervous system and the peripheral nervous system. White axons happen only when the neuroglia are wrapping the axons here. So it's in the central, oops. The term, instead of a continuous action potential, it's called a saltatory action potential. Saltatory means jump. It goes from one spot to another and to another, and I'll talk about what that is in a second. The axons themselves propagate this action potential very, very rapidly. When I was in graduate school, there was a bunch of us eating lunch one day, and I had my back to someone, a friend of mine who was had an orange, and she thought I was looking at her, and she goes, "Hey, Mike," and I turn around, and I see this orange thing all I see is this thing coming at me, and I go like this, I caught it right there. And I looked like a damn ninja. It was really cool, right? But I had no idea she was throwing it, and everyone was like, "Oh, you do that again, do it again." I was like, "No, it's never happening again." But how did that happen? In the time that I turned around, I had no idea what was happening. I see this orange thing coming at me, and then I had to react to it. So now, the signal is coming in my eye, it goes to my brain, from my brain, it goes to my deltoid, my biceps brachii, my brachialis, and then I catch it, right? So it goes to, and sends signals, all those. That's how fast that has to happen, right? That is an example of saltatory action potentials here. And being more specific here, saltatory means jumping, and so the action potential goes from one node to another to another. The AP jumps So in the drawing that I made in the previous diagram, I talked about it being the node of Ranvier. The node of Ranvier is the area where is unwrapped axon that exposes the axolemma, and it exposes the axolemma to the action potential. So the axolemma here is the exposed axon. Where the action potential occurs. So there's two different types of action potentials that occur on different axons, and they're either continuous or saltatory, and the saltatory ones are much more interesting. They're much faster, they're much longer distance, and they can happen in the brain, in the spinal cord, and also in the periphery. Continuous action potentials do not happen in the periphery. Those neurons are just too short, so they happen solely in the central nervous system, either in the spinal cord or in the brain. Okay, so what I want to do now is let's take a break, and I want to switch devices here, and I want to just show you